Pfizer-BioNTech COVID-19 vaccine could neutralize Brazil variant, lab tests show. As new variants of the COVID-19 virus emerge, vaccine manufacturers are testing their products against them. Recently, a study published in the New England Journal of Medicine revealed that the Pfizer-BioNTech vaccine worked efficiently against the Brazil variant. However, the vaccine's performance versus that new variant was only confirmed in lab experiments. According to Reuters, the study was conducted by scientists from Pfizer and the University of Texas Medical Branch. Their paper noted that the Brazil variant or P.1 lineage exhibited mutations in the S gene of the coronavirus, similar to the UK and South Africa variants. These mutations altered the spike protein, enough to resist antibodies of survivors and vaccinated individuals, as presented by other studies. To test the vaccine against the Brazil variant, they engineered S mutations from each of the three new lineages into an early isolate of SARS-CoV-2, which was from January 2020. They produced three recombinant viruses representing each of the lineages and two extras that were engineered to have the subsets of mutations of the South Africa variant. Overall, three individual recombinant viruses, each represented all mutations of one variant, were created and tested. Next, they used 20 serum samples obtained from 15 participants in the pivotal trial of the Pfizer vaccine. The samples were gathered 2 or 4 weeks after participants received the second dose of the vaccine at 30 micrograms. All serum samples neutralized the January 2020 virus isolate without a hitch. The samples also neutralized all viruses with spikes from variants. Performance-wise, researchers compared the neutralization activity of antibodies between the isolate and the variants. This showed them that the neutralization against the UK and Brazil variants was about the same as the neutralization against the isolate. But the neutralization against the South Africa variant was slightly lower compared to others, which is found as well in previously published studies. While the results are encouraging, researchers cautioned about the interpretation. They said that the study was limited to the mutations altering the neutralization via changes in the spike protein. They couldn't tell if the results would be the same in antigenicity or the ability to be recognized by antibodies during an immune response. Thus, real-world evidence is a must to solidify the findings in lab tests. Once evidence is available, it can show if the antibody or T-cell activities induced by the vaccine truly work outside controlled settings.